Okay, hello Biochem. My voice is starting to come back, so hopefully this one goes a little bit smoother than the last one. So this is day 2 of fatty acid oxidation. Here are the learning objectives. State the cellular location for beta oxidation. Draw the structures for the intermediates of all 4 reactions. State the type of reaction catalyzed for each pathway, and the chemical logic for the first step in beta oxidation. Okay, so our fatty acid oxidation, or otherwise known as lipid catabolism, there's a few other kinds of lipids that we could talk about, but we're only doing fatty acids. Like, we're not going to do how you make a cholesterol or anything like that. So last time we did how you get the fatty acid from the adipose tissue or from when you eat like a stick of butter into the bloodstream. That's through the chylomicrons and then lipolysis into the cell. And then the fatty acid is activated and then it's transported into the mitochondrial matrix. And so today we're talking about beta oxidation, how we start to actually break down the fatty acid. And then our last day of fatty acid oxidation is going to talk about some nuances. What happens if you have double bonds or cis bonds or a really long chain. And we're going to include some of the diseases associated with fatty acid catabolism. All right, let's get it. Okay, so beta oxidation is a pathway. It's a process that occurs in the mitochondrial matrix. And it has 4 steps. And each round, you're going to get out 1 acyl-CoA. And the reason we're going to get out 1 acyl-CoA is because every round of beta oxidation is going to chop off, yikes, chop off, it was better the first time, 2 carbons at a time. And our 2-carbon unit that we have in the mitochondrial matrix is the acetyl-CoA. So every 2 rounds, we're going to get, the chain is going to get shorter by 2 carbons because we have 2 carbons leaving. There's 1 carbon here and 2 carbons there. 2 carbons leaving as acetyl-CoA. If you notice, we're not going to get any CO2 out of this. So if we want to completely oxidize the fatty acids, this acetyl-CoA is going to need to go to the TCA cycle. Okay, let's see. So these are the 4 basic steps here, and they have a general outline that is oxidation, hydration, oxidation, and then lastly is thiolysis. And we're going to go through each one of them because you have to know the structures for all of the intermediates. I'm going to give you the enzymes for these, but you need to know the structures and the names of all the intermediates. So we're going to go through them, and then we're going to put it all together and combine it with all the other metabolic pathways that we've learned. In the packet that I gave you, you should have this diagram that you can go ahead and fill out with me as we go. I'm going to zoom in and zoom out because the view that I have right here is too tiny. Like, I'll never be able to get it all. But we're going to fill in all these boxes. Okay. So what I want you to notice is that even though the intermediates, the carbon metabolite intermediates, are named here, you are going to need to have those memorized. Okay, so we're going to start out with an acyl-CoA, and actually I'm going to draw this going from right to left instead of left to right. So I'm going to put the CoA over here. There's my thioester bond. And then I'm going to draw out the first 2 carbons, or actually the first 3, really, because the carbonyl is carbon number 1. And then we're going to have the rest of our chain of the acyl-CoA. And then, of course, we're going to end it with the CH3. And we're going to do 16 carbons total. So there's going to be 12 methylene groups in the middle there. And I'm going to draw out the hydrogens for this. Okay, beta oxidation means the beta carbon will be oxidized. So coming off of the carbonyl, this is alpha, and this is beta. So in biochemistry, oxidation means that we add an oxygen. So you're going to be watching for that beta carbon to be oxidized. So when in doubt, sorry, I'm trying to drink orange juice while I'm doing this. When in doubt, when you're trying to decide where to put the oxygen, put it on the beta carbon. Okay, so this is called an acyl-CoA because the number of carbons coming off of the CoA is more than one. If there's only one, it would be called acetyl. Okay, so the first step, remember, is our oxidation. Step. And what that means is we're going to remove some hydrogens. So the acyl-CoA will be oxidized to trans delta 2 enoyl-CoA. All right. Oh, I gotta zoom in. So the hydrogen, so the name here tells you a little bit about what type of double bond we're going to make. So we're going to make a trans double bond, and delta 2 is from your symbolic notation, so you know that the double bond is going to be on carbon number 2. So I'm going to zoom it in here. No, I'm going to zoom back out, sorry. So we're going to be losing, let's take this hydrogen, and we'll put them on an FAD. Will be reduced to FAD, and I'll just keep the colors H2. So we're going to lose those two hydrogens, are going to be placed on an FAD to make FADH2. And the enzyme here is acyl-CoA dehydrogenase. Again, all the dehydrogenases are named for their substrates. So acyl-CoA, we're doing an oxidation reduction, so it's a dehydrogenase. Okay. Zoom, zoom, zoom. CoA, carbonyl, carbon. And I'm just going to put an R group here so I don't have to keep drawing it out. I got to look back and see which two I took off. So put my double bond here. Okay, so that's my trans delta 2 enoyl-CoA. The hydrogens are on opposite sides. The other groups are also on opposite sides. Trans delta 2 enoyl-CoA. The second step is hydration. Okay, it's exactly what it sounds like. We're going to be adding a water. I'm going to use red, and I'm going to draw out my water molecule. And I'm going to just we're going to add the water molecule across the double bond. So I'm going to put a couple different shapes here. So I've got like a cloud shape over the OH and a triangle over that H. So we're going to add across the double bond. So here's where we're adding our oxygen. And hopefully you can at least guess that the oxygen is going to go on the beta carbon because this is beta oxidation. So you oxidize the beta carbon. Zoom, zoom, zoom. CoA, Thioester, carbon, carbon, R group for just the rest of the thing. And I have to check and make sure. Okay, so I'm going to add the OH here, and the other hydrogen from water is going down here, and then I have my originals there. And this is beta-hydroxy-acyl-CoA. Again, beta for beta carbon, hydroxy means OH. And of course, acyl-CoA means acyl-CoA. All right, so now the next, it says the words right here. Hilarious, I've been writing them out. So it goes oxidation, hydration, oxidation. So the next step is, again, oxidation. And what colors am I using for that guy? And this is where we're going to get an NADH to come out. And the two hydrogens that we're going to lose to be this one and this one, because again, we're oxidizing the beta carbon. So I'll do NAD, I'll put the red hydrogen there and the yellow hydrogen here. So what's going to be formed is a carbonyl. Okay, so I've taken both of the hydrogens off of the beta carbon, so that carbon is now completely oxidized. The only thing that it has on it are oxygens. And this one is a CH2. I'll just keep my colors the way that I've been going. And this is beta-keto acyl-CoA because you have a ketone at the beta carbon. All right, and then the last step is cleavage, also called Thiolysis. And here's where we're going to get our acyl-CoA to come off, and we're going to shorten the chain by 2 carbons. So all right. So this CoA is going to come in and attack the beta carbon. Oxygen's up, oxygen's down. This is actually going to break a carbon-carbon bond. We're not going to actually talk too much about that, but I'm just going to grab this hydrogen so that you can see right here is the acyl-CoA that is leaving. Okay, so this acyl-CoA is what is leaving. And that was from carbons 1 and 2. Oops, there's a bond there. I've got hydrogens coming from all different places. Okay, that's the acyl-CoA, and we've shortened the carbon chain, the acyl chain, by 2 carbons. This is carbon 1 and carbon 2. So that what I've got now, again, is another acyl-CoA, but it's 2 carbons shorter. So again, I'm going to draw it here, CoA, S, carbonyl. I'll draw the next 2 carbons, the methylene, and end it with a CH3. So this is beta oxidation only for saturated fatty acids, some things with no double bonds. And because we've lost 2 carbons, this is now only 10 carbons long. So we started with something that had 16 carbons, and if you don't believe me, it's 12, 13, 14, 15, 16. And we've lost 2 carbons, so now we're at 10, 11, 12, 13, 14 carbons. And the process was to go around and around and around and around and around and around again until all you have left over are just a big pool of acyl-CoAs. So again, the process is oxidation, hydration, oxidation, thiolysis. Oops. And each one round, you get 1 NADH and 1 FADH2. Okay? And I want to talk a little bit about the number of rounds and the number of acyl-CoAs that are coming out here. So instead of drawing every single step out, we're going to do a fatty acid that's 16 colon 0, so 16 carbons long, no double bonds, and it's an acyl-CoA. So that's carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, so we're going to talk about number of rounds and number of things that are coming off here. So every round we shorten by 2 carbons. So this is carbon 1, this is carbon 2. So we would cut here. Carbon 1, carbon 2, cut. So each dotted line is a round. I guess I could just make them regular cuts. So again, I'm cutting 2 carbons off every time. And what you'll notice is that the final round has 4 carbons. 1, 2, 3, 4. And the final round will give you 2 acyl-CoAs. So when you have 16:0, the number of rounds is 1, 2, 3, 4, 5, 6, 7. So number of rounds of beta oxidation equals the number of carbons, the number of carbons divided by 2 minus 1, because the very final round you get 2 acyl-CoAs out instead of just 1, because we have 4 carbons. You cut that in half and you get 2 acyl-CoAs, which means that the number of NADH equals the number of rounds. And so in this case, the number of NADH we have is 7. And the number of FADH2 also equals the number of rounds. Let's do my little, it's the same thing. And we get 7 of those. The number of acyl-CoAs, though, is a little bit different. So if you notice, we have 1, 2, 3, 4, 5, 6, 7, 8 acyl-CoAs. The number of acyl-CoAs is the number of carbons divided by 2. So in this case, we have 8 of those. And this is not the completion of oxidation of a fatty acid, because we haven't made any CO2. So remember, we need to get all the carbons, all the carbon-hydrogen bonds to turn into carbon-oxygen bonds. And so the acyl-CoAs will go into the TCA cycle. So we're going to draw that out so you can see the connection of these pathways. All right. One of these days you're going to get tired of seeing me draw this. All right, this is the matrix. This is the cytosol. This is the outer membrane of the mitochondria. This is the inner membrane, and this is the intermembrane space. It's the space in between the two membranes. And I'm going to put CM here for cytoplasmic membrane. All right, let's review again. Glucose. Glucose can come from glycogen or from when you eat it. Hey, come on. Let me erase that. Okay. Glucose comes in through a transporter and is oxidized partially into pyruvate. And then that pyruvate can do, some of the pyruvate will enter into the mitochondria, and some of it will be used in fermentation to make lactic acid. Glucose to pyruvate is called glycolysis. Pyruvate to lactic acid is called lactic acid fermentation. And of course, out of this we get we have 2 of those, we have 2 of those, we have 2 of those. And this regenerates NAD+ to keep glycolysis running, at least for a little bit. The pyruvate that winds up in the mitochondrial matrix will be partially oxidized into acetyl-CoA, our good friend, which has 2 carbons, so we know that we lost a CO2, which is what I mean by partially oxidized, because pyruvate has 3 carbons. Again, we get NADH out, and we had to put a CoA in. And then to complete the oxidation of acetyl-CoA, it goes to the TCA cycle. And the final 2 carbons come out as 2 CO2s. So that's the complete oxidation. Again, we get 3 NADH, 1 FADH2, and 1 GTP per turn, per single turn. When we have a chylomicron show up with the tags inside of it, the tags undergo lipolysis and are broken into glycerol and 3 fatty acids. The glycerol will enter sideways into glycolysis, so we don't waste any of those carbon-hydrogen bonds. And the fatty acid will first undergo activation. I gotta zoom in here because this is gonna be really teeny tiny. To become acyl-CoA because it cannot cross the membrane on its own. This is gonna cost an ATP. I'll put the arrow in. We're also going to need a CoA. The acyl-CoA cannot cross the membrane, so in order to transport it or to shuttle it across, the CoA is swapped out for a carnitine. So we get acyl-carn, I'm just going to call it carn, and the CoA comes off. And carnitine goes in. Acyl-carnitine is able to be transported. And once inside the mitochondria, is reverted back to acyl-CoA. And again, carnitine comes off, and a CoA goes on. All right, so we have lipolysis followed by activation, followed by transport. So now we have our acyl-CoA in the mitochondrial matrix. And I'm going to do this for beta-oxidation, where it goes around and around and around and around and around again. And what comes out is acetyl-CoA. These are the carbons, so notice all of my carbon intermediates and carbon metabolites are all in blue, and all of my reduced coenzymes and ATPs are all in green. Each round, we get 1 NADH and 1 FADH2. If this acyl-CoA was 18:0, I would get 9 acetyl-CoAs, and I'd get 18 divided by 2, which is 9, minus 1, which is 8 of these, and 8 of these. And of course, I'd have to put some CoA in, a whole bunch of CoA, 9 CoAs. No, 8 CoAs, because I start with a CoA. Okay, and then this is the reason that beta-oxidation occurs in the mitochondrial matrix, because the completion of catabolism requires these acetyl-CoAs to enter into the TCA cycle and spin around and around and around and around again. So if we have 9 acetyl-CoAs, the TCA cycle will spin 9 times. So if we want to go from a fatty acid to CO2, and eventually, of course, we're going to add water or oxygen and water to our combustion reaction. In order to go from fatty acid to CO2, we have, let's go back here. We have activation plus transport plus beta-oxidation plus the TCA cycle. Whoops, TAC. Crazy. TCA cycle. Analogously, if we have glucose being oxidized, to CO2. And again, eventually this is going to be a combustion reaction. In order to get glucose to CO2, it's glycolysis plus the PDC, or a bridge step, plus TCA cycle will get us to CO2. Okay, so we're going to do the last little bit here is we're going to do some ATP math. And I'm going to show you how to write it out so that you'll get full points for showing all of your work. Okay. Let's go. I don't know why I'm just moving it around. Let's just add a new slide. All right, so let's say we have, let's do 18. No, let's do, 18:0 fatty acid. All right. First of all, in order to do activation, this is going to cost us an ATP. So I'm just going to subtract 1 ATP from activation. Okay? And then the fatty acid will be transported into the mitochondrial matrix, and then it will undergo beta-oxidation. So from beta-oxidation, what we're trying to find here is total number of ATPs produced. Okay. So from beta-oxidation, we know that for an 18:0, the number of rounds is going to be 18 divided by 2 minus 1. So that should be 8 rounds. In each round, I get 1 NADH and 1 FADH2. So if I have 8 rounds, for every 1 round, I get 1 NADH. So that will multiply out to be 8 NADH. And again, 8 rounds. For every 1 round, I have 1 FADH2. So that equals out to 8 FADH2. Next bit of math that you'll need to know is that for every 1 NADH, it equals 2.5 ATP. And for every 1 FADH2, that equals 1.5 ATP. I gotta do the math. Maybe you can get your calculator out and do the math while I'm doing the math, or we're going to pray that my calculator works, right? Because I don't want to have to remake this video. So this should be 20 ATPs. Excuse me. And this is 12 ATPs. And just so you know, we will actually talk about the pathway that this comes from and the pathway that this comes from. This is electron transport chain plus ATP synthase, which is they both are, both in green here, which is sometimes called oxidative phosphorylation or OXPHOS. Okay. Hey, squiggly line, go away. All right. So we have, from beta-oxidation, we have our NADH and FADH2 accounted for. And then we know number of acyl-CoA equals number of carbons divided by 2. So that's 9 acetyl-CoAs. And the acetyl-CoA will go into the TCA cycle. So if I have 9 acetyl-CoA, for every 1 acetyl-CoA, I get 3 NADH from the TCA cycle. And again, we know that 1 NADH is worth 2.5 ATPs. And again, 9 acetyl-CoA. So I know that this seems tedious, but this is the kind of logic that I'm looking for you to state when you do your problems. I want to see that you can show all of the steps. And then please, please, please don't forget about GTP because it's the equivalent of an ATP. Go away. 1 GTP, and every 1 GTP is the equivalent of 1 ATP. Okay, time to do some math. Let's see, okay, 9 times 3 times 2.5. Oops, hey, go away. What's the matter? 9 times 3 times 2.5. Whoa, that's so many ATPs. Yeah, that makes sense. It's a lot of ATPs. 9 times 1 times 1.5, 13.5. And again, we'll also talk about this 0.5 or this half of an ATP. We'll talk about that later. And then this is going to be 9. Okay, so in order to find the total number of ATPs, you're just going to add them all up. So I have, let's see, 9 plus 13.5 plus 67.5 plus 12 plus 20. Did I miss any? Oh, minus 1. Does that seem right? Did I miss any? I got minus 1, 20, 12, 67.5, 13.5, 9. Wow. Hey. Okay, so my grand total is 121 ATPs. Whoa, that's a lot. So if you remember that glucose will give us about 32 to 36 ATPs. You can see that a fatty acid will give you a ton more ATPs, and that's because it has a lot more carbon-hydrogen bonds. Okay, let me check my notes. You'll be doing this type of problem a lot. So please, please, please show all of your work. Show all work. And what I mean by work, I mean this is what I want to see, is all the logic, all the steps laid out, all the stoichiometry laid out. Please, please, please don't skip any steps. You can even label, this is coming from the TCA cycle, and oxidative phosphorylation. This is beta-oxidation and oxidative phosphorylation. And then again, don't forget, you lose an ATP for activation. Okay, I think that's all that I have. Happy ATP math hunting, and I'll see you in class. Bye-bye.